Okay, hello everyone. Thank you for being here with us in another great webinar. And thank you for joining us in our past webinars. So today we have another great talk by Kalpana Raja from Morgridge Institute for Research. So the title of her talk is SKiM, a generalized LBD system for uncovering discoveries from PubMed. So thank you, Kalpana, for your participation in our webinars. We are really happy that you are being part of this initiative. So the floor is yours, and thank you again.
Thank you so much. Hello, everyone. Today I'm going to present Serial KinderMiner, or SKiM in short, a generalized literature-based discovery system for uncovering unknown biomedical discoveries from PubMed. Currently, PubMed includes more than 31 million articles, and more articles are added to the database every day. The approach used is text mining. As a traditional approach, we have to keep on reading the articles one after the other. This is going to be tedious, time consuming, and impractical. Alternatively, we can use the text mining approach, which is automated, quick and efficient, and accurate. So, let me say how text mining can be done. As I mentioned already, PubMed includes more than 31 million scientific literature, the published articles. The first step is to get the literature that is related to our research. This is called information retrieval. Let me say an example. If I'm doing a research on Alzheimer's disease, then I will be interested in the literature published on Alzheimer's disease. So, getting the subset of the literature related to the Alzheimer's disease is called information retrieval in text mining. So once we have the subset, the other steps in text mining come into picture. The most common next step is information extraction. Here, we automate to extract the information from the literature. So we write computer programs to achieve the task. The information could be anything depending on the user's demand. I'm just giving few examples here. Gene-gene interaction, gene-disease association, gene-chemical interaction, gene pathway association, and there are many more. Once we have the information extracted, either we can go and build the knowledge bases so that it will be available publicly to other researchers. Some of the examples include GSEA and CTD. Otherwise, we can integrate the data together for hypothesis generation that finally leads to knowledge discovery. So before I enter into the topic, I want to share the lexicon compiling. I use the lexicons in my research. I compiled 3 different lexicons, one for the disease, the next for the drugs, and the third one is on phenotypes and symptoms. The disease lexicon was compiled from Eumelus metathesaurus and SNOMED CT. It includes 640,000 diseases and synonyms. The drugs lexicon is compiled from Eumelus metathesaurus, Drug Bank, and Form GKB. This lexicon includes more than 1 million drugs and synonyms. So it's a very comprehensive list. In addition, I compiled the phenotypes and symptoms lexicon from 3 available resources: Human Phenotype Ontology, PHEOS, and OMIN. So once I combined all the information from the 3 resources, I introduced an approach to remove the prefix and suffix related to the medical terms. This approach gave me new phenotypes, but it included some false positives as well. Then further, I included the synonyms for the phenotypes from Eumelus metathesaurus and SNOMED CT. Finally, I carried out the manual evaluation to remove the false positives, that are the wrong informations on the phenotypes, which I got from the step prefix and suffix. So this finally gave me a list of phenotypes and synonyms. Finally, I applied one of our text mining tool called KinderMiner to find the phenotypes and the synonyms mentioned in the literature. So this is my phenotypes lexicon. So before getting into SKiM, I would like to share the previous tool. Before SKiM, we developed KinderMiner, so I would like to share some information on KinderMiner. KinderMiner reads 30 million PubMed articles just in minutes. So here I'm just giving a comparison. Just imagine printing off all the biomedical papers in PubMed and piling them up one above the other. So this is going to be as high as 23.3 miles high. This is 5 times higher than Mount Everest, whose height is 5.5 miles. So now you can compare. Is it possible to read all the articles manually? Definitely not. So KinderMiner is an alternative way. This is a text mining system. It reads and summarizes the knowledge from 30 million biomedical papers just in minutes. So KinderMiner narrows the experiments for the wet lab scientists. There are so many diseases, drugs, genes, small molecules, and organisms. Everything together will lead to millions of lab experiments. So KinderMiner can narrow down this to tens or hundreds of lab experiments. So now I'm entering into today's topic, literature-based discovery. So let me give a brief introduction on literature-based discovery. In short, we say LBD. The overwhelming size and rapid growth of PubMed articles is a challenge for the researchers to keep themselves up to date with the literature published in their own specialization and to know the discoveries made in other domains. LBD uncovers undiscovered public knowledge by finding the association between the two topics that are otherwise unrelated. Don Swanson pioneered LBD. He found dietary fish oil for Raynaud's disease. LBD can be of two types, closed LBD and open LBD. In closed LBD, the inputs are A and C terms to find the intermediate common B terms. In open LBD, the input is just the A term. First, it finds significantly associated B terms for the A term. Then, for every B term, it finds the significantly associated C terms. Thus, the C terms are associated with the A terms via the intermediate B terms. In specific, A and B terms are present in a particular set of PubMed articles. B and C terms are present in a different set of PubMed articles. So sometimes they are unrelated. So we get the new discovery by connecting two sets of PubMed articles. As I mentioned already, today the talk is on serial KinderMiner or SKiM. This is a LBD system and it is an open LBD system. SKiM is a generalized system applicable to process a variety of biomedical queries. So here I present the workflow based on its application on drug repurposing. For this application, the A term is going to be the disease of interest. It could be any disease. B term is a list of phenotypes from our lexicon. And C term is going to be a list of drugs. Again, it is from our lexicon. So first, SKiM finds AB association. It gets the significant B's associated with A. Then it sorts the B's depending on a prediction score. Then we take only the top n B's where n stands for number. It could be any number. So for every B from the top N, SKiM finds the associated C terms. Finally, it groups all the B terms for every C term, then sort it based on the prediction score. So here I provide the example for this drug repurposing application. Raynaud's disease is going to be the A term. B term found by SKiM is blood viscosity, and C term found by SKiM is dietary fish oil. So blood viscosity is a condition observed in Raynaud's disease. Dietary fish oil reduces the blood viscosity. Thus, dietary fish oil is a remedy for Raynaud's disease. When Swanson identified this, he found the information from two different PubMed articles that were unrelated. So that time it was a new discovery. Any LBD system is commonly evaluated on two approaches. The first one is to rediscover the discoveries made by Swanson and his colleagues. The second one is a time slicing approach. So we also evaluated SKiM on both the evaluation approaches that are commonly in use already. In addition, we also compared SKiM with KinderMiner. So on the left side, I'm listing the list of discoveries made by Swanson and his colleagues. So let me take the first one, Raynaud's disease and dietary fish oil. So here, we allow SKiM to read the articles published up to a certain year. So this year, we call it as cutoff date. So we want SKiM to find the discovery by reading the articles up to that year. So initially, we gave the cutoff date as 1 year prior to the discovery made by Swanson and his colleagues. SKiM identified or rediscovered all the discoveries made by Swanson and his colleagues. KinderMiner found only one discovery. Then we relaxed the cutoff date to 2019. Even then, KinderMiner was able to find only 3 discoveries and failed to find 2 discoveries. Thus, SKiM is more sensitive and more powerful than KinderMiner. Here, I want to mention that KinderMiner can summarize the information from PubMed articles, and it is not a LBD system. But SKiM was developed based on the features from KinderMiner. That's why we made a comparison with KinderMiner. So now let me explain the second evaluation approach, that is on time slicing approach. So here we give the year or the cutoff date to allow SKiM to read the articles up to that particular date to make the discovery. Then we compare it with the first clinical trial or the first landmark publication. So let me explain this with the same example, Raynaud's disease and dietary fish oil. Swanson and his colleagues made the discovery in 1986. So we allowed SKiM to read the articles up to the year 1985. Swanson's discovery was first validated in the year 1989. So this was the first clinical trial. This means SKiM is able to find the discovery 4 years prior to the first clinical trial, confirming the usefulness of the drug for the disease. It is same for the rest of the discoveries as well. So in addition to the existing evaluation approaches, we also evaluated SKiM findings on our drugs and disease association. This we compiled it from 4 existing resources, namely Comparative Toxicogenomics Database, or CTD in short, National Drug File Reference Terminology, or NDFRT in short, Drug Bank, and Form GKB. We calculated recall and precision. Again, we compared the performance of SKiM with KinderMiner. The The first cutoff date is 1 year prior to the discovery by Swanson and colleagues. So in case of Raynaud's disease, this is 1985. The second cutoff date is 2019. As you see here in figure A, SKiM achieved better recall than KinderMiner on all the 4 diseases in both the cutoff dates. So here RD stands for Raynaud's disease and AD stands for Alzheimer's disease. The 4 diseases we considered in our study are from the Swanson's discovery: Raynaud's disease, migraine, Alzheimer's disease, and schizophrenia. So the next step is to find the precision. KinderMiner and SKiM finds all possible significant associations from the literature, and this is going to be a very vast list. Our DDA, the Disease Drug Association, is an expert curated one. So there is a big gap between the expert curated one and whatever is published in the PubMed, because the expert curated one is a manual approach. So sometimes the true positives that are available in PubMed are not going to be in the expert curated list, so that will affect the precision. So a common approach is to find precision at n hits. So here again n stands for the number. It could be any number, the top n hits. We took top 20 hits to calculate the precision. So this I marked it as precision at 20 in figure B. So it is the same set of diseases: Raynaud's disease, migraine, Alzheimer's disease, and schizophrenia. Here I carried out 3 different levels of evaluation. I marked them as E1, E2, and E3. So E1 represents precision at 20, that is based on evaluation using only DDA. So it's very low. Then we took a manual analysis to find whether any information is a top true positive in PubMed. So that improved the precision. This is E2. So in addition to E1 and E2, we also took a manual analysis across the PubMed articles to know whether the finding is a true positive. So here, this is marked as E3. So again, there is an improvement. E2 and E3 are both manual analysis, but with a slight difference. In E2, I looked for PubMed articles that clearly says the usefulness of the drug for a disease in an article. So it is just A to C. In E3, I looked for articles on A to B and B to C. So this is A to B to C, so totally new discoveries. So this is the summary on the repurposed drugs for all the 4 diseases at 2 different cutoff dates. So as I mentioned, it got a big list. So I just got this box plot to get the most potential and promising drugs to the top. So these drugs are marked as D. And I compared the new drugs with the known drugs. So the known drugs are shown in green color box, and the next one is the new drug, and on the top I'm showing the most promising new drug candidates for further analysis in the wet lab and clinical trials. So just as an example, I want to show the hits for Alzheimer's disease using the cutoff date as 1995. So I'm showing only the top 20 SKiM predictions. So here, the most interesting hit is metformin. This is a known drug for type 2 diabetes. Recently, it is found to be useful for treating Alzheimer's disease as well. So we gave the cutoff year as 1995 to make this prediction. The first clinical trial validating metformin for Alzheimer's disease happened in the year 2017. This means SKiM is able to find the discovery 22 years prior to the first clinical trial. This is cool, isn't it? Also, the first literature indicating the usefulness of the drug for the disease was published in the year 2011. This means SKiM is able to find the prediction 16 years prior to the first literature indicating the usefulness of the drug. So it is possible to get some idea on future discoveries using LBD systems like SKiM. So in addition to the 4 diseases I discussed so far, we also applied SKiM on repurposing drugs for 22 conditions with high prevalence rate in USA and worldwide. This includes type 2 diabetes, kidney failure, stroke, asthma, and the complete list is here. So this is what we found. It's a lot of information on the slide. So I just want to summarize. So the recall is always better for SKiM compared to KinderMiner. So I'm just enlarging only 4 diseases: bipolar disorder, hypercholesterolemia, Parkinson's disease, and psoriasis. So in all the 4, you see that the recall achieved by SKiM is better than the recall achieved by KinderMiner. So the next one is the precision at 20 for all the 22 conditions. Again, it's a lot of information. So the take-home message is E2 is better than E1 and E3 is better than E2. We got good precision at 20 for all 22 conditions. And here is the repurposed drugs for all 22 conditions. So most of the drugs are associated with a disease with a very weak prediction score, especially the new drugs. So those were pushed to the bottom. Only some of them appeared with high prediction score. So those things I marked as D. These are the most promising drug candidates for further analysis in wet lab and clinical trial. So I want to share type 2 diabetes from the 22 conditions. The cutoff date is March 2019. I'm showing only the top 20 predictions made by SKiM. The most interesting one is treprostinil. This drug is found to be useful in treating type 2 diabetes, and it was published in the year 2020. We made the prediction in March 2019. So SKiM was able to make this prediction 1 year prior to the first literature indicating the usefulness of the drug, as well as the first co-occurrence of type 2 diabetes and treprostinil in the same PubMed article. So here you also see a lot of false positives. This is because the current version of SKiM does not differentiate between the positive and negative associations between A, B, and C terms. In future, we will address this issue with advanced NLP approaches such as BioBird or similar ones. We are working towards that. With this, I end my talk today. I have a lot of people to acknowledge from my current lab, especially my mentor Ron Stewart and my colleague John Style. I also thank all the funding agencies. So here I'm sharing the picture and you see our entire group here. Thank you so much. I'm ready to take the questions.
Okay, thank you, Kalpan. A great talk. So we have time for questions. Let's wait a couple of seconds because we have a delay with the YouTube broadcast. Let's wait for questions if there are any questions. But in the meantime, I would like to ask you. So you mentioned that SKiM finds statistically significant, for example, as you say, A-B pairs from one set of PubMed articles and BC pairs from a second set of PubMed articles. This is a kind of guilty by association, maybe, that you can infer that those terms are related or are associated. So have you think to include in your software a graph module, maybe if you implement kind like networks in order to display the results that comes from a SKiM? I mean, because if you use networks, you can have the big picture of the relationships among different terms. And also it will be really interesting to find the hubs. I mean, the terms that are related, that are critical with the network in different kind of abstracts. Have you thinking in implement this kind of graphical resources?
It's a great idea. So currently, SKiM is available as a command line tool. We already put it in GitHub. Also, we are developing the web interface. That is the current project I'm working on. So your idea is great, and I will think about it. So if I present it as a graph, it'll be easy to navigate and find the information. That's a great idea. Yes. Thank you so much.
Yeah, that will be really nice, and I think that will be the big difference from your development with other kind of text mining tools, because the majority of the text mining tools sometimes run in a command line in a standalone version. Maybe a web interface will be helpful for different kind of people, for different kind of professions. Physicians, maybe, why not? Maybe in the future physicians can use your software, or any other kind of people that are related with with health sciences. So let's wait. I think that we have no questions, but I have another one. For me, was a really interesting talk from you. It's a really nice software that you have developed there in your lab. So have you applied this scheme to find in terms of the repurposing drugs and SARS-CoV-2? Have you tried with SARS-CoV-2?
Yes, we tried. We tried, and by the time we got the results, especially we are trying to publish SKiM before we go ahead to publish the results on COVID-19. So it's kind of little delay, and now the vaccines are coming out. Still, we are trying to publish that paper too, and we are getting very interesting results using SKiM.
Yeah, that will be useful to apply this tool. It is also because you have data from protein-protein interactions, host-parasite interactions, and maybe you can integrate those PPIs with your results, with the skin outcomes. So that's really nice, Kalpana. Thank you.
Also, we applied SKiM on repurposing phytochemicals. This is in collaboration with the JSS College of Pharmacy from India. So the further idea is to take the results to the wet lab experiments. So we just collaborated with them and got the SKiM results for them. So they are trying to come out with an idea. So when the vaccines are ready, they are trying to find whether any of the phytochemicals could be an adjuvant for the vaccine. So that's what they are focusing. So already we use SKiM for repurposing phytochemicals.
Awesome. Congrats, Kalpana. It's a really
thank you
amazing job. So thank you, Kalpana. I think that we have no more questions. For me, it was a great talk. And we are so happy to have this kind of talks that are really interesting developments in the field of bioinformatics and computational biology. So thank you all for being here today with us, and thank you again, Kalpana. And you are very welcome to back here in our stage to present your future results.
Definitely. So this is the second time I'm presenting my results in ISCB-SC, and I'm looking forward for more opportunities to present my future results as well. Thank you so much for the opportunity. Thanks.
Okay. Thank you. Bye.
Bye.